Perché siamo in diretta streaming con Corriere TV e dobbiamo partire puntuali. Grazie mille a tutti, grazie.
Good afternoon. My name is Alice, and I'm a member of We Science, the youth group of Bergamo Scienza. Before starting, I'd like to get your attention, reminding you that connecting to the website bergamoscienza.it and clicking on the live button, you can follow the event in streaming directly on the YouTube channel of Bergamo Scienza and also on Facebook page. You can send questions to your speakers or invite your followers to follow the conference live. The questions from the web are selected by us, the volunteers, and these will be asked during the question and answer session. The conference of the 22nd edition of Bergamo Scienze are made accessible to people who are disabled, both sensorially and motor perspective, and there is a sign language service. All the conferences will be video recorded. Some will be also streamed live. If there are foreign guests, simultaneous translation will be provided and the recordings will be published on our sites. And all these services are made available thanks to the Fondazione della Comunità Bergamasca and Fondazione Banca Popolare di Bergamo. Together with Bergamo Scienze, they want to promote inclusion and accessibility. For simultaneous translation, you can scan the QR code that the volunteers in the room can provide you with, or you can find projected behind me. The QR code will allow you to download the Commercio web app, where you can select the interpretation language from the drop-down menu. To listen to the translation, you need earphones. Please use your own. If you don't have one, please ask the volunteers in the room. I would like to welcome you to the closing event of the 22nd edition of the festival, titled mRNA, from anti-COVID vaccines to the fight against tumors. We welcome Drew Weissman, Nobel laureate for medicine 2023, and there will be also the mayor introducing the conference. We also have a visual thinker who will be taking a graphical recording of the meeting. Thank you very much for listening and enjoy your meeting. Thank you. We can start this last session of the Bergamo Scienza Festival. It was a very interesting festival. It is a great honor for me to introduce and interact with Professor Drew Weissman. It is a great honor to close the 22nd edition of Bergamo Scienza Festival, Science Festival. This Science Festival is an outstanding opportunity we are provided because over the past few years we could celebrate scientific curiosity, better understand how the world works around us, and also be surprised. And allow me to say that we learned what we can bring home, take away from knowledge, from sharing, so as to try and understand what can be the solutions to present and future challenges. We are privileged today to have with us Professor Drew Weissman, who is Nobel laureate for Medicine 2023, together with Professor Katalin Karikó. The Nobel Prize, as you might know, was awarded to him given his key contribution to the development of mRNA vaccines. We are specifically referring to COVID-2019 vaccines.
Grazie.
Thanks to this discovery, thanks to these vaccines, we could save millions of human lives across the world, and we could go back to our normal lives, as you can see here in this room. Professor Weissman was born in Lexington. There he grew up, and since when he was a young child, he got interested in science. And his father was an engineer, and he worked in the production of optic mirrors for satellite use. And this experience brought Weissman to get closer to science and to technology as such. He studied biochemistry, enzymology at the University of Brenda, where he got his master's degree in 1985. He attended Boston University. He got a degree in medicine. He had a PhD. And his dissertation was on microbiology. Since 1990, Professor Weissman accepted a scholarship to work at the National Institute of Health in Maryland, and it was there that from 91 to 97, under the guide of Anthony Fauci, he focused on the immunopathogenesis of HIV. In 97, he went to School of Medicine of the University of Pennsylvania in Philadelphia. And after then, he started working on what brought him to the Nobel Prize. He started studying dendritic cells that play a key role in the immune surveillance, and he focused more and more on the development of mRNA vaccines. The idea of using mRNA for vaccines and the administration of live viruses was something that was put forth in the 80s, but before this could become a reality, the problem was to overcome some technical obstacles and even scientific ones. The first experiments indeed showed that mRNA transcribed in vitro prompts inflammatory responses and an inefficient production of proteins, both in cells and tissues. After some time, after starting working in Penn, Professor Weissman met Professor Karikó and shared his interests on mRNA to prompt the body to develop immunity against pathogens and viruses, and they started collaborating. This was a turning point in the advances of mRNA vaccines development, and it was a joint discovery of both of them, showing that mRNA produced by modifying the molecules can avoid the recognition of inherent immunity. It is from this discovery that two scientists could work on the new innovative technology, which was safe and effective for vaccine development. When in 2019 COVID started spreading and the Bergamo area was badly hit, there were very few people that we could develop vaccines that could avoid the spread of the virus. And indeed, very many vaccinations were approved, and two of them were produced thanks to the new mRNA technology. And it was Professor Weissman and Professor Karikó's discoveries to make the vaccine platform of mRNA suitable for clinical development when it was mostly needed. And it is for this reason that we all know that this was the most outstanding contribution to human development. But there is more to this. And this is the reason for this conference, from anti-COVID vaccines to the fight against the tumors, because very many other diseases can be treated by means of these developments. Professor Weissman and his team are focusing their research to develop a vaccination that is effective against coronaviruses and a universal vaccine against influenza, and also keep fighting tumors. So it is a great pleasure for me to welcome Professor Drew Weissman in Bergamo at Bergamo Scienza Festival. Thank you very much, and the floor is yours. Thank you.
Thank you very much. It was a wonderful introduction. Mayor Carnevali, thank you very much. Thank you for the organizers for inviting me here to speak today. So what I wanted to talk to you about today was a few different things. I wanted to start with the vaccine, but what I'm going to talk about is why the vaccine works so well. What's the mechanism? Not a lot of people know this, and this hasn't been widely spread with the vaccine. The second thing I'll talk about is the future of mRNA. What are the potential applications, therapeutics, cures that can be developed using this new technology? And the final thing I'll talk to you about is equity in vaccines and therapeutics. So this is the timeline for mRNA development. I put this up because when the pandemic first hit and the vaccine was released, I spent a lot of time talking to lay audiences, trying to convince them why the vaccine was safe, why they needed to take it. There was one thing that every audience asked me, and that was they would say, well, I don't know if I want to take it because it was invented in 10 months, and I think that was too fast. This timeline shows you that it wasn't invented in 10 months. mRNA was first discovered in 1961, long before most of you were born. The first time it was injected into animals and to people was in the 1990s. This isn't a brand new technology. What Katie and I did is we made it work better, and we made it work as a vaccine. Now, after it was first studied, it was only used for vaccines, but with a very unusual vaccine. What they would do is they would take dendritic cells, cells that are part of the immune system, out of mice and then out of people, pulse them with mRNA and give them back. That didn't result in any curative effects, It didn't help treat cancer at all. And most biotech companies, pharmaceutical companies, and others gave up on RNA and said, it doesn't make enough protein, it's too inflammatory, it's too hard to work with, and they gave it up. And everybody dropped mRNA. Katie and I started at that point in time, and we were struck by how inflammatory the RNA was. And that was surprising to us because every one of our cells is full of RNA. And the RNA we were giving, we thought was identical to the RNA in the cell. So why would the body see that RNA as inflammatory? As it turns out, there are 17 different proteins, sensors, that recognize RNA in our cells, in our bodies. They're everywhere. They're in extracellular fluids. They're on the cell membrane. They're in the cytoplasm. They're in the endosomes. They're on mitochondria. And they do lots of inflammatory things. They make inflammatory mediators. They induce apoptosis, which is a form of cell death. So Katie and I came into it at this point in time, and we wanted to understand why RNA was so inflammatory. We spent about 7 years working on this, but this was one of the telling experiments that gave us an idea of what was going on. And what we did here, we isolated different kinds of RNA from mammalian cells and bacteria. These are from different organelles, different regions, different types of RNA. And we added each of these RNAs to dendritic cells, which recognize inflammation and recognize RNA. What was striking was that one of the RNAs, transfer RNA, had little to no inflammatory activity, while bacterial RNA had a lot of inflammation. So what does this mean? What it involves is nucleoside modification. So nucleoside modification is the changing of parts of RNA. RNA is made up of 4 nucleosides, 4 letters. And those letters code for all of the proteins that our body makes. But our cells modify those letters to have different functions, to regulate a variety of things. They're conserved. So a bacteria has the same modification in a ribosomal RNA that we have in our ribosomal RNAs. The thought was then, how could we make modified RNA to test? Would that make a difference in inflammation? So what we did is we figured out how to trick a phage polymerase. That's an enzyme that copies DNA and makes RNA copies. It usually uses the 4 nucleosides. We tricked it into incorporating a modified nucleoside. So what that means is that every U is now a pseudouridine, or every U has a 5-methyl modification. And when we tested those modified RNAs on different types of dendritic cells, and we saw that any U modification had a reduction in the amount of inflammation of the RNA. That was wonderful. We proved that the source and the way to overcome inflammation was by modifying the RNA. But the next experiment was what was truly fascinating. We wanted to know, could the RNA that we modified still make protein? The function of RNA is to take a code in our DNA, copy that code, bring it to a machine known as a ribosome that reads the code and makes protein. We weren't sure whether modified RNA could still be translated. Well, we were wrong, and we were wrong tenfold in the wrong direction. Not only does the RNA still make protein, but it made much more protein. So that addressed two of the big concerns that pharmaceutical companies and biotechs had: inflammation and not enough protein. We also found that the RNA had contaminants in it, and if we got rid of those contaminants, we could increase the amount of protein made by 4 orders of magnitude. That's a huge increase in the amount of protein. So it's around this time I'm still going to HIV meetings. I'm still an HIV researcher. And I would sit down with Tony Fauci, Gary Nabel, other leaders in the field, and they would ask me what I was up to. And I would sit down and I would explain to them the work we were doing on RNA, and they would smile and they would nod, and often they would say, well, you know, Drew, you really should drop RNA. It's a useless field, it's never gonna work, nobody's ever gonna use it, you're wasting your career. Why don't you do something useful with your life? And I would argue with them and say, well, you know, there's a lot of potential for RNA. This is how proteins are made for therapeutics. They're in 50,000 liter drums full of CHO cells. And then you have to figure out how to purify the protein. Every protein you start over. That's why monoclonal antibody therapies in the US cost $50,000 to $100,000 a year. RNA can do that in a much simpler manner. We're the source of the protein production. Our cells, the RNA just gives the code. So it's more effective, it's safer, and much less expensive. You just had to solve all the problems with mRNA. The next problem, though, was delivering the RNA. Every cell and every fluid in our body has enzymes in it that digest RNA, known as RNases. So if you don't protect the RNA, if you don't cover it, if you don't hide it, the body destroys it in seconds. We studied around 50 different kinds of formulations: lipids, sugars, proteins, peptides, alginate, whatever we could find. We finally got our hands on lipid nanoparticles. These were developed to deliver short RNAs, 21 base pair double-stranded RNAs, and they're part of FDA, EMSA-approved drugs. We wanted to know, would they work for mRNA? So we made an RNA that encoded firefly luciferase. Firefly luciferase is the enzyme that fireflies have that makes them glow at night. That enzyme acts on a substrate and it produces light. We can do the same thing in a mouse. We can give them the RNA encoding the enzyme and then look where the light is. What we found is that if we gave it systemically into the blood, it went to the liver, which is what it's supposed to do. But when we injected it like it was a vaccine into the muscle, into subcutaneous, into intradermal, it stayed at the site of injection, and it was translated. It made protein for over 10 days. From a vaccinologist's point of view, that's great. In general, when we make vaccines, there's lots of different types of vaccines. We call them platforms. Live virus vaccines, that's the measles, mumps, and rubella, and a few others, work the best. They give the most potent immune responses. The reason we don't like to develop them is they're attenuated viruses, and sometimes the attenuation can disappear, and you get pathogenicity back. So the FDA, EMSA, other organizations don't like live virus vaccines. The advantage of a live virus, though, is it produces protein for many days, which is important for making a potent immune response. So now we had an RNA that produced protein for 10 days, and we were excited to see how well it worked. This is just a simple representation. You don't need to know the specifics, but what you need to see is that we're comparing an inactivated virus, a live virus, both influenza, so the old folks in the audience get an activated virus, the young people get live virus in their nose. We compared those two types of vaccines to an mRNA encoding the influenza antigen. What we found was that the mRNA was 50 times better than the inactivated virus and 5 times better than the live virus. In general, pharmaceutical and biotech companies get excited when you have a vaccine platform that doubles the titer of an immunization. They were very excited by this. We wanted to understand how the vaccine worked. So this is just a cartoon that shows you all the steps that a B cell, which is the cell that makes the antibody, goes through to respond to an antigen. We measured these responses in an animal. And what we did is we did this in an antigen-specific manner. So what that means is we added the hemagglutinin protein with a fluorescent tag. And then we measured how many of each type of B cell were there after a single immunization. And we found that for all 3 cell types, there was about a 50-fold increase in the number of antigen-specific cells. So that explains the high titer. We forgot about some mice. We left them in the colony for over a year. I'll talk to the people about that later. But we went back and we studied these mice to see, was there any immune response left? And we looked in the bone marrow, because the bone marrow is where the plasma cells, the cells that make antibody that appears in our circulation, live. We found an enormous number of hemagglutinin-specific plasma cells in the bone marrow. So we developed a new vaccine platform that used a combination of mRNA and LNPs together. It was incredibly potent. One immunization protected animals against challenge with a variety of viruses. But we had an issue. We didn't understand part of this. And the main thing we didn't understand was, where is the adjuvant? All vaccines have an adjuvant. An adjuvant stimulates the immune system and gets it to work better. Vaccines have to have an adjuvant in it in order to work well. I showed you that the RNA had no adjuvant activity. It made no inflammation. And the lipid nanoparticles weren't known to have any adjuvant activity either. So we couldn't figure out where was the adjuvant that was making this vaccine work so well. There are many different kinds of CD4 helper cells. We focused on one type of cell known as a T follicular helper cell. This is a helper cell whose only job is to make germinal centers. Germinal centers are where antibody responses develop. The stronger the germinal center response, the better the antibody response. So we measured these T follicular helper cells. And we did these in a macaque who was immunized with the HIV envelope, part of an HIV vaccine. And the reason we did this is that we had an identical protein antigen similar to our mRNA LNP that we could compare. And this antigen, we gave it the best adjuvant we had for making T follicular helper cells. But as you can see, there's no comparison between the mRNA and the best adjuvant around. We did some calculations. About 5% of the CD4 helpers are TFHs with protein and RNA. With mRNA LNPs, it's over 50%. So this vaccine specifically induces T follicular helper cells and produces protein for a bunch of days. We wanted to know then, where was the adjuvant? The LNP shouldn't have had it. The mRNA didn't have it. We did a simple experiment. We took empty lipid nanoparticles and added them to hemagglutinin protein and compared that to mRNA with the same protein encoded in it, and the LNP turned out to be the adjuvant. When we compared this adjuvant to other types of adjuvants, we saw that it was very different. It made different kinds of responses. It induced different mediators. The mediators it did induce are ones associated with T follicular helper cell generation. So that was good. My lab, since the COVID-19 vaccine, has continued to work on many different vaccines. We mainly focus on vaccines where previous vaccines have failed. Things like HIV, hepatitis C, herpes simplex, norovirus, C. difficile, EBV, influenza, pan-coronaviruses, and many others. We're also working on vaccines for other diseases. One of them are food allergies. So any parent in this room who has a child with a peanut allergy knows what a nightmare life with that child can be. Always afraid if they go to school or a friend's house and they eat something with peanuts, they end up in the emergency ward. We figured out how to make a vaccine that will block those allergies for peanuts, for all foods, for aeroallergens. Those are things like dust mites and pollens. We're working on vaccines for autoimmune diseases. Currently, the main treatments are immunosuppressive, things that turn off the immune system. And they help, not 100%, but they can help. The problem is they make people at risk of developing severe infections or cancer. We're working on vaccines that only turn off the immune response to the autoantigen, the antigen that causes the disease. And we're also working on a variety of different cancer vaccines and different types of cancer vaccines. So the standard vaccines that people think about are, after you've been diagnosed with cancer, you have an operation, they try to take out as much cancer as they can, and then they give you chemotherapy or immunotherapy or a vaccine to get rid of any leftover tumor. We're also looking at another form of cancer vaccine called an intercepting vaccine. This is a vaccine for people who have been identified with a genetic marker that leads to cancer. The BRCA genes are the most well-known, but there's many of these. These are genes that many of them associate with the development of cancer at an age range that varies. The idea of this vaccine is you give it before you ever find the cancer. And it makes the immune system get rid of any malignant cells before they can turn into a real cancer and cause difficulties. We're also interested in targeting the LNPs. So I told you before that LNPs go to the liver. That's what they were developed to do. But what if you wanted to treat something else? What if you wanted to treat brain or heart or lungs or T cells or bone marrow stem cells? So we figured out how to target the LNP to switch it from liver to other cells and organs. We did a very simple procedure to do this. We added an antibody to the LNP surface, and the antibody would bind an antigen associated with a different cell, tissue, or organ. When we did this, the LNPs didn't change morphology. They got a little bit bigger, but otherwise they stayed the same. Most importantly, they still worked. So you can see in C, these are human endothelial cells. And without targeting, they don't take up RNA LNPs. With targeting, and we're using a PCAM molecule that they express, you get high-level uptake. So specific uptake in a specific cell type. We injected these targeted LNPs into a mouse. You can see on the top, without targeting, most of them go to the liver. With PCAM targeting, over half of them now go to the lung. The lung has the highest expression of endothelial cells that express PCAM. So we were able to switch where these LNPs went to, and we can now deliver therapeutic RNAs to the lung. We also wanted to see, could we target the brain? I'm a physician as well as a researcher, and we had what we called the Friday night nightmare. And this was a patient who was admitted on a Friday evening with a new stroke. And you can see in the CAT scan on the left, there's an area of black brain. That's the region that the stroke has killed the brain. This patient likely could walk in. They might have a bit of a speech impairment, but they were otherwise healthy. The problem is what happens over the next few days, and that's on the right. And as you can see, the area of dead brain has now nearly taken over the entire hemisphere. That's because dead brain releases inflammatory mediators. That causes the blood vessels to leak, the inflammatory cells to come in, and the size of the stroke to greatly increase. The person on the right likely didn't leave the hospital. So we developed a way to simulate this. We would inject an inflammatory mediator into one side of the brain, and we delivered an LNP that was targeted to the brain. And we put an mRNA into it that encoded an anti-inflammatory mediator. So these were given intravenously into the vein, and the stroke was put into the mouse's brain. What we saw is that with treatment on the far right, in the absence of treatment, there's high levels of release of fluid from the blood vessels, vascular leakage. But with a delivered anti-inflammatory to the brain, we've returned this to baseline. The CAT scan of the mouse no longer showed expansion of that stroke. We're now looking at this in clinical trials to see, can we improve people who have strokes or other brain insults? As an immunologist, I've had a long-term interest in modifying T cells. T cells do a lot of important things. They're involved in fighting off a lot of different types of infections. They're involved in helping to control tumors and other malignant processes. if we can modulate T cell functions, we can make them work better, make them kill better. We can stop their killing. So there are diseases where the immune system is too aggressive. We can turn that off. We can deliver specific molecules that give them new functions. There's a lot of flexibility of what you can do to a T cell to make it work differently. The first problem, though, is can we deliver an LNP to a T cell? And in general, we were unable to do this until we targeted it to the T cell. So an untargeted LNP, you can see in blue under purified CD4 cells, had no activity, wasn't taken up, didn't make any protein. But when it was targeted, we efficiently delivered to CD4 positive T cells. We injected these LNPs into a mouse, and again, what we saw with CD4 targeting, the spleen lit up. The spleen has around 20% CD4 T cells. But what was more interesting is we re-imaged the mouse after we removed all of those organs. And what lit up were lymph nodes. So lymph nodes are what collect the lymphoid drainage from all of our tissues. What this means is that the IV injected LNPs escaped from the circulation, went into the tissues, into the draining lymphatics, back to draining lymph nodes, where they still could bind CD4 cells and deliver mRNA. That told us that we had a therapeutic approach. We used a set of mice that essentially had a built-in marker where we could deliver a protein that edits their genome only where the RNA is delivered. When we did that, on the y-axis, you can see high levels of fluorescent activity. That means the gene has been edited. But most important, what this also shows you is that the x-axis is an activation marker. 90% of our T cells are resting, unactivated. And that's most important for HIV because HIV sets up long-lived reservoirs in resting CD4 positive T cells. So our hope is that we could target CD4 T cells and deliver a therapeutic to latent HIV as an alternative form of cure. What we saw from this is that we targeted resting CD4s as efficiently as activated. So we could gene edit a resting CD4. We now have experiments where we're delivering a variety of enzymes that inactivate, that cut, that kill latent HIV provirus in cells as a way of asking, can this be used as a form of cure? About 50% of the CD4 positive T cells are targeted with a single injection. So we figured out how to now target the LNPs. And importantly, they maintain function. They retain the ability to deliver the RNA to the targeted T cells. You can use this for a variety of therapeutic purposes. You can put gene editing machinery to fix mutations in genes. You can deliver a variety of stimulatory or inhibitory therapeutics. Penn is well known for developing CAR T cells. The issue, though, is you have to be able to deliver to all T cells. So we studied other markers of T cells and found one called CD5 that delivered to all T cells. You can see on the right, 80% of CD4 and CD8 positive T cells are targeted with a single injection. So what a CAR T cell is it takes our natural CD8 positive killer T cells and turns them into specific killers. So our T cells recognize antigen associated with MHC. And why that's limiting is most of us have a variety of different MHCs and a variety of different peptides. So you can't use this approach for everybody. But what a CAR T cell does is it makes a synthetic T cell receptor that recognizes a tumor-associated antigen. The first ones that were developed recognized CD19. CD19 is expressed by all B cells. It's also expressed by leukemia and lymphoma cells. CAR T cell treatment is about 60 to 70% effective. Now, these are on patients that were going to die quickly from their disease. So that's an incredible cure. The problem with CAR T cells is what it takes to make a CAR T cell. You have to leukapheresis a patient. What that means is you put them on a machine that takes out blood, spins it, removes the white cells, and gives everything back. And it does that over and over and over for hours. You then take those white cells and you infect them with a lentivirus, a virus similar to HIV that can deliver a new gene. And that's how you deliver the CAR gene. So that makes the cells produce the CAR. Then you have to expand those cells for about 10 days to make enough cells to give back to the patient. Then you have to give the patient chemotherapy. So you have to kill off T cells to make room for all the T cells you're adding back. That results in fairly high morbidity and some mortality. Our idea was that since we're now able to target T cells, could we target a T cell and deliver a CAR to that T cell encoded in the mRNA? That way, T cells in the body would take up the LNPs, express the CAR on their surface, and kill whatever the CAR was directed at. We used the model of cardiac fibrosis. And in this model, you give the mouse drugs that give them high blood pressure. And high blood pressure leads to cardiac fibrosis. This is echocardiogram measurements. It's not important to know what they are, but what you should realize is that the gray is normal animals. The yellow is hypertensive animals. You can see that the volume of the heart increases. The amount of blood it can pump is reduced. Red is treated once with mRNA LNPs that deliver the CAR molecule. It returns the heart function to normal. So this is an off-the-shelf drug. You take a vial of RNA LNPs, you thaw it from your freezer, and inject it into a mouse, and someday into a person. We stain the heart muscle for fibrosis that shows up in red. On the left is a normal heart, very little red. In the middle is a hypertensive animal, lots of red fibrosis. And on the right is a single treatment with the FAP-CAR LNPs that return the fibrosis almost to normal. We also have a big interest in bone marrow stem cells. So this is important for a variety of reasons. There are thousands of immune deficiencies, red cell deficiencies, and other congenital immune defects from bone marrow mutations. The classic is the SCID baby. So these were immune deficient babies that in the old days were put in bubbles because they had no immune system. Nowadays, the principal disease that we think about is sickle cell anemia. 300,000 people a year are born with sickle cell anemia, mainly in sub-Saharan Africa, but across the entire world. There is a new treatment that was approved last December of a gene therapy for sickle cell, where they take out a lot of bone marrow cells, add a lentivirus that fixes the gene, give them chemotherapy, and give them the cells back. The problem is it costs $3.2 million per person, and it can only be done in select centers that have the knowledge and the facilities to perform these series. If you multiply 300,000 times 3.2 million, that's an unacceptable number. So we wanted to see, could we target bone marrow stem cells? Now what this is showing you is, if you look at the numbers, if you start with 700,000 bone marrow cells, less than 1,000 are actually stem cells. So they're incredibly rare and hard to find. We developed a different kind of targeting. We used a marker known as CD117 that's present on repopulating stem cells, as well as other cell types. And with a single treatment, we could target 80+% of the stem cells. We treated mice and studied them 6 months later. These are mice that have that fluorescent protein that's turned on by a gene editing enzyme. What we saw is that after 6 months, 70+% of the cells remain gene edited. We then took the bone marrow from that mouse and did a bone marrow transplant into a new mouse. And the new mouse also had 70+% transformed cells. So that means a single injection into a vein could modify, could gene edit the repopulating bone marrow stem cells. And we're now moving forward to treat sickle cell anemia. My dream someday is to go somewhere in the world, sub-Saharan Africa, India, anywhere, with an igloo cooler, a frozen cooler full of RNA LNPs, and line people up and give them a single injection of RNA LNPs to cure their sickle cell anemia. There are many other uses you can do with bone marrow targeting. You can deplete bone marrow stem cells when you want to do a transplant. You can correct genetic defects. You can deliver absent proteins, and you can deliver therapeutic proteins that make more granulocytes, platelets, red cells, other things. So I look at the future of mRNA as in vivo delivery, being able to fix things in the body, not having to take cells out, not needing millions of dollars of fancy facilities to be able to fix things. And that leads to my interest in equity. So I've always had a long-term interest in vaccine and therapeutic equity. This means getting the new therapeutics, the new vaccines, available to the entire world, not just the wealthy nations. This is the current world population as of 2015. As you can see, the majority of people are in sub-Saharan Africa and Southeast Asia. This is the distribution of money in the world. The majority of the money is in North America and Western Europe. This is the distribution of malaria. It doesn't match where the money is. It matches where the people are. And what this means is that you have the poorest people in the world suffering from malaria, TB, and a variety of other infectious and non-infectious diseases, these people can't afford the fancy drugs that we develop in the US and Western Europe. So how do we get them access to these drugs? I've been working with a group in Thailand for almost 10 years. Well before anybody knew what mRNA is, we started a collaboration where I helped them develop their own mRNA vaccine research. I trained all of their investigators. I worked with them every week to think of new ways, new treatments, new vaccines, new developments. Around March of 2020, Kiat in this picture, who I work with, who runs the VRC, came to me and said, we're concerned that any vaccine that's made in the West, in the US or Western Europe, won't make it to Southeast Asia or Sub-Saharan Africa or Latin America for years. And they weren't willing to wait. So Kiat went to his government, went to the royalty of Thailand, went to charitable donors, and got enough money raised to make their own vaccine. So Kiat and his people and my people worked together to make our own mRNA LNP COVID vaccine. This is the phase 1 clinical trial of that vaccine. We're comparing it to two things. In the green, that's the Pfizer vaccine. And in the purple, that's people who have had the disease within the past month. As you can see, the Thai vaccine is better than the Pfizer-BioNTech vaccine. I work with BioNTech. I say that very slowly when I talk to them, but the vaccine worked better. Their next problem was producing the vaccine. So when a vaccine or any drug is made, it's made under what's known as Good Manufacturing Practices conditions, GMP. This is an incredibly expensive, highly complex production system that's required to make drugs or vaccines. Thailand didn't have one before this. So working with Kiat and with the funders, we were able to build a GMP production site in Thailand where they're now producing their own vaccines. They have 10 different vaccines in clinical trials. Now, why this is important from an equity perspective is two things. One is the next time a pandemic hits, and I can't tell you if it's going to be in 1 year or 100 years, but there will be more. Now, Kiat and Southeast Asia will have the production system up and running to make their own vaccine. Working with the WHO, we've built 15 more GMP centers across the world in Latin America, Sub-Saharan Africa, Southeast Asia, Eastern Europe. We actually built one in Ukraine in the middle of the war. We've also built a hub in South Africa outside of Cape Town. And the purpose of this hub is to teach anybody in the world who wants to learn how to make mRNA LNP vaccines and therapeutics, how to do it. They'll show you all of the equipment, all of the procedures, all of the analyses. They'll train your people. They'll give you help in anything you need. So this allowed the 15 GMP sites to be built and to be running. So I need to thank all of the people in my lab. These are the people who actually did the work. And I need to thank all of the other labs that I've worked with over the years to accomplish everything that we've done. Thank you very much.
Grazie.
Thank you, Professor Weissman.
Credo che
I
l'abbraccio
believe that this warm rounds of hand is showing great recognition because the first thing I think of is good thing that you did not to take the advice not to go ahead with your studies on RNA, because we heard why these vaccines work, why they are safe, and what are the underlying mechanisms for their working. But on the other side, we also know
applicazioni che sono in corso.
what are the ongoing applications. I was deeply struck when you referred to many autoimmune diseases or food allergies or the post-stroke edema, which is very important because the consequences can disrupt the brain functioning and can kill you.
Così come
And also, you mentioned BRCA disease, and you also spoke about the evolution of studies on bone marrow stem cells, and you have the merit of delving on equity. That was a huge problem during the pandemic period, and we realized that we were on the side of the lucky ones, those who had the opportunity to have access to vaccination and drugs. And thanks to your teams, the laboratories you are collaborating, that the equity approach allowed for greater justice, because this is very important in any civil society. It is difficult to start asking questions. I have, out of curiosity, a short question. At a certain point, you said, I am a physician beyond being a researcher. And out of curiosity again,
specializzarsi.
amongst the various disciplines you could have got specialization. Why did you opt for vaccination? Why you decided to deal with vaccines? And I assume that you would do the same thing again should you start from scratch.
So to be completely honest, vaccines are considered low-hanging fruit. They're the easiest thing to develop when you create a new platform. As an immunologist, I've always been interested in vaccines, but we made a vaccine in 10 months. If we were to make a new therapeutic, that would take much longer. So I've always been interested in vaccines as an immunologist, and I'm glad we were able to do it so quickly.
Bene. Sembra che arrivino delle domande invece.
Come sempre alla fine della conferenza abbiamo spazio per le domande del pubblico. Ci sono i nostri volontari in sala, quindi se avete una domanda per il professor Weissman, alzate pure la mano.
Should you have any question, the volunteers can come and bring you the mic so that you can ask your question to Professor Weissman.
Buonasera.
Good evening.
Avrei voluto fare la domanda in inglese.
I would have liked to ask a question in English, but I was told I need to speak Italian. I have a question.
In esecuzione degli studi.
Are there studies being carried out
dei risultati positivi per quanto riguarda i vaccini contro l'herpes virus? E se invece non ci sono o non sono
and if there are no ongoing studies or if there is results are not positive, why is it so against HSV?
So you were asking about HSV, herpes simplex virus. So we have a phase 1 clinical trial that's just about completed. The results so far look good. The code hasn't been broken, but we see very good immune responses in half the people. So we're now doing the control, so are they efficacious? We should know the answer in probably less than a year.
Abbiamo anche una domanda dal web. Silvia da casa ci chiede che suggerimento può dare a una biologa
researcher biologist who is interested in studying oncology towards a more targeted medicine, well beyond any kind of challenge.
So the field of oncology has probably changed more than any other element of medicine. When I was a resident, just training physician, about 80% of patients who were diagnosed with cancer died. Nowadays, it's less than 20%. We've made really enormous progress in treating cancer. To me, what's interesting, though, is the future of cancer treatment is now going to be directed at the cancer cell. So in the old days, you gave a toxic drug, and you hoped you killed the cancer before you killed the normal cells. Nowadays, most new treatments are directed directly at the cancer and only kill the cancer cells. As that develops, it gives you more power, more strength in the treatment, and better tolerability. And both of those are important.
Abbiamo un'altra domanda.
There is another question here, first row.
Thank you so much for your talk. I had a question about, if any, what role artificial intelligence is playing in your lab, or if you see it play a role eventually.
So I often will get into arguments with people about artificial intelligence. The way I view it is it has the capacity to remember hundreds of thousands of structures, of chemicals, of pieces of data, and incorporate all of those into a treatment. We use AI as a way of optimizing RNA structures. So RNA is made up of a cap, UTRs, a coding sequence, a poly A tail. Each of those has to be optimized. You can either do it by random guessing, or you can use AI to try and pick what's likely to work the best, which is what we do.
C'è una domanda a metà sala.
There is a question down there.
Grazie.
Thank you. My question might sound trivial. Why is it that vaccines aimed at preventing diseases are also used as therapeutics to treat?
So it's often that therapeutic vaccines and prophylactic, preventative vaccines target the same organism, but they induce different kinds of immune responses. So for herpes simplex virus, to prevent infection, the vaccine makes antibodies. To control infection, the vaccine needs to make cellular immunity, T-cell responses. So there are some times when therapeutic is the same as prophylactic, but often for chronic viruses, they're different. For human papovaviruses that cause cervical cancer and different oral cancers, antibodies prevent infection, cellular immunity cures disease.
May I, out of my curiosity, given what you said during your presentation, speaking about intercepting vaccines and referring to BRCA gene diseases, you said it is important for this opportunity to use the vaccine is before you actually discover the tumor. In this case, it is possible because you first map the gene. In other cases, how could this be possible?
So this is the future of medicine. At last year's Nobel Prize, the chemistry laureates, we got into discussions about new ways of detecting disease. So right now, you bring your child to the pediatrician when they have a sore throat, they culture their throat, and if they've got streptococcus, they give them antibiotic. But the future is very different. What the future now looks like is you walk up to a machine and you wave your hand over it, or you breathe on it, or you give some saliva or other bodily fluid to the machine, and the machine can analyze hundreds of thousands of components and give you with very high accuracy, you might be very early for colon cancer, stage 0, no detectable disease anywhere. But the machine will be 90 to 95% correct that over time you will develop colon cancer. So my hope is that we develop the intercepting vaccines and then combine it with that kind of technology where you don't need a gene defect or a gene mutation. You just go into your doctor, you wave your hand over the device. It says, you're going to get diabetes, you may get colon cancer, you may, whatever. And then we can start treating them when they're phase 0, before they really start to develop, instead of waiting until you have a big tumor and surgery and all of those difficulties.
Ci sono altre domande dalla sala?
Are there further questions from the audience in the room?
Sì, buongiorno.
Yes, good afternoon. I'd like to refer back to what you said. You already partially answered therapeutical vaccines against the tumors, and I would like to refer back to what you said about AI. In melanomas, algorithms have already been used to forecast the coding of anomalous proteins that are coded by triplets that do not work well. But the use of AI in tumor microhabitats where both healthy and tumor cells communicate, can this kind of communication be useful to better modulate the immune system so that it's more effective?
No, certainly. So I think AI will be very important in selecting antigens to put into a vaccine. Nowadays, the most current vaccines that have the best success are what's known as personalized vaccines, What they do is that they take a piece of the tumor from the patient and they sequence the genome of the tumor. They identify all of the mutated proteins, and then they have AI-driven algorithms that tell them which of those mutated proteins are likely to give a potent protective immune response and which ones aren't, and which ones are likely to cross-react with normal tissues and which ones aren't. So it helps them formulate what to put into the vaccine to get the best efficacy and the least amount of adverse events.
Abbiamo una domanda anche qua.
One question here, fourth row.
Parliamo sempre di prevenzione.
We keep speaking about prevention, Professor Weissman.
Perché molte persone ancora oggi
Why is it that many people today are fearful, do not want to get vaccinated?
We would need many hours to discuss that. And I'm a basic scientist, so I don't particularly understand why people are so fearful. I think people have always been fearful of new medical treatments. The smallpox vaccine goes back to 1796 or so. People were fearful then. There are pictures in magazines of people being drawn with horses, with cows' heads coming out of their ear or out of their hand. So even hundreds of years ago, people were fearful of new treatments and new vaccines. The hope is that the scientists can explain why it's a good treatment and why it's safe. The difficulty that we've run into during the COVID pandemic is it became a political exercise to speak badly and to combat against vaccines as a way of gaining power and gaining money. So So people are using it as a way of dividing and as a way of, hurting other people to make their own, get their own power and money.
To close, I'd like to ask Professor Weissman if he has any tip to give us young people like volunteers of Bergamo Scienza Festival who are looking at their professional career in scientific research, who would like to start a job in science. Have you got any piece of advice? What are the characteristics that we should be developing to be a successful researcher or scientist?
Yeah, in general, there aren't particular characteristics or traits that define a researcher. One of the important things is curiosity. It's inquiring into understanding what you don't understand and trying to develop better. I tell young people all the time, if they share that, then science is a great place to go. It's a job where you can reinvent your job whenever you want. If you suddenly want to investigate something else, if you want to study a new type of cancer or a new virus, or if you want to develop new computer programs or new robotics, science is the way you do that. And you have the flexibility that you can do just about anything you want once you've finished your training and you've got the support of your fellow researchers.
Grazie.
Grazie a tutti.
Thank you very much. We have come to the end.
Grazie mille alla nostra sindaca Elena Carnevali.
Thank you to our mayor, Elena Carnevali. Please remain on stage for some time more, because I'd like to thank a lot of people for this festival. What I'll take away after this festival is that we understood from our speakers that we are exploring nature. We have understood that nature already provided us defense systems in 3 million years, because otherwise we wouldn't be here interpreting them, using them, because they are much more powerful than chemical drugs we developed in the past. The side effects are much less. We got the possibility to learn these things from Nobel laureates, but not only during this festival. This is our takeaway message. And to young people, biology is going through very many changes. You can make a difference. Look for new solutions. There is plenty of room to do science and to do it well. So I'd like to thank now my wonderful volunteers that over the past 15 days
che ci fanno sperare.
Gave us hope, because they worked hard to welcome you all, to welcome our speakers. You know that our organization, association, is volunteer-based. We do this because we love science. And also this is a very important moment for our association. We plan to turn into a foundation. Hopefully we can do this, and hopefully all our supporters and volunteers can take part in this initiative, because we want to keep organizing this festival, not just in Bergamo. It should become international. It should be local, but then become global. It should be available to you all free of charge. I also would like to thank the founders, organizers, supporters. I don't want to mention any name because I would definitely forget some. I would like to thank those who attended and spoke. Hundreds of people followed us, and this is very exciting to know. It was a very successful edition, so we are very happy. You know that there is a right, so I asked the Professor Weissman to remain on stage so that we can take a picture with all our supporters and organizers and volunteers. So without stealing any more of your time, also because we want to keep in our mind what Professor Weissman told us about vaccination and the good tips he gave to young researchers. So all the volunteers are kindly invited to come on stage, please. All the founders, Mario, Raffaele, Andrea, Alessandro, and also Matteo, Paola, Katia, Paola Locatelli, Nives, Nicola. You are all invited on stage.
Tutti sul palco. E con questa foto di rito veramente vi vogliamo ringraziare.
So we really would like to thank you for being so numerous. Thank you very much, and long live to Bergamo Scienza Festival.
Grazie ancora a tutti e buon Bergamo Scienza.